Hello and welcome. I'm Professor Beams with the ACC Landscape Horticulture Department, and this is the first lecture in the Propagation and Greenhouse Operations series. Now, a lot of folks will start taking this class for a number of reasons, one of them being having interest in a potential career in a greenhouse or nursery environment, or even wanting to start their own either commercial or personal greenhouse. And another reason is there's such a satisfaction in being able to grow your own plants, either from cutting or from seed. It's nice to support our local businesses and go buy fully mature plants from a nursery or a greenhouse, but it's also really nice to be able to know how to do it yourself. It also gives you some important insight into how plants grow and what proper care actually looks like. And my example that I want to give for that right now is this little friend we have right here. So this is a ghost plant or Echeveria succulent. And typically when we see these, well, they grow in a couple of different colors. Sometimes they're red, sometimes they're purple, sometimes just green. Sometimes there's all three of those colors present in one plant. But typically when we get that plant first from the store, we buy it for ourselves, we receive it as a gift. All of these shoots grow as these tight little clusters. And so they are usually fairly low to the ground, and they've got sort of this cute, chubby, compact look to them. And that's the desirable form of the plant. But over time, they just start to become kind of leggy. And I've had a lot of friends and family and students ask me, well, what am I doing wrong? It's growing too tall. Is it water? Is it light? Is it the pot? And the answer is no, that's how it grows. But each of these little lobes is its own potential plant. And so what I want to bring your attention to is this one right here. Now, I promise I didn't knock this plant around. These lobes are just barely hanging by a thread. So we'll go over this in another lecture for sure. But if you just pull one of these off, you can see, if you look really closely, there's a little bit of a nub on the end right there. And so what we do is we're just going to lay this right down on the surface of the soil. We're going to make sure that the plant has as much water as it needs, but over time, and we'll see this in subsequent lectures because I'll keep you posted as it grows, this little guy will turn into its own new plant and its new little cluster. So point being, when we take this course, we are going to be learning a lot about how plants reproduce, how to identify the different types of reproduction that a plant is going to use, and we're going to figure out how to make that work for us so that we can have a really healthy, continuous growth of our plants. Beginning with the definition, plant propagation is the process for multiplying or increasing the numbers of a plant species. And as we mentioned, there are several different methods available to propagate different plants. Now we can see here in this image of a greenhouse, perfectly uniform plants growing at the same rate, and they look almost identical to one another. So when we start talking about the different methods of propagation, we're going to start to see that there are different ways to either clone a plant so that we literally have genetically identical propagates from the parent plant, or then we can start to get into the fun part about hybridization and finding new ways to combine the DNA of different plants to try to get some of the more desirable characteristics. There are two types of propagation. Sexual propagation is by seed, and we call it sexual propagation because it actually involves the sex organs of a plant, which are flowers. So we have the male parts of a flower and the female parts of a flower. And when they come together, when they love each other very much and come together, they create seed. Or there is asexual propagation, and that is by cloning, which is what I was demonstrating with the ghost plant. So another term that is going to come up is parent stock. So the parent stock is whatever plant material was used to create the next generation of plant. So the parent stock could be the male and female plants that were used to make a seed, or like I demonstrated with the lobes of the ghost plant, it could be plant material that is used to clone the original parent stock into new stock. So to reiterate, asexual propagation is the process of growing new plants from parts of an existing plant. So the different plant parts available to us are stems, buds, leaves, roots, and there are also specialized plant structures that are used for propagation. These are going to be tubers, rhizomes, bulbs, plantlets, and stolons. So this is an example of using a leaf to propagate. This is an example of a tuber. And yes, I did have to pull this off of the top of the compost pile because I realized that I needed it for this video. Should have eaten it. And then this, oh, where is it? This is an example of a plantlet. So I've got a large aloe vera plant and then a little baby aloe vera plant popped up right in there. Plantlets also can be referred to as pups, and I think that in this area of Central Texas, that's what they're commonly called, especially with aloe vera and aloe vera's cousin, the agave plant. In my opinion, rhizomes are one of the most interesting plant parts that we can use for propagation. So we call these rootstocks or sometimes creeping rootstocks, and you know it because it's out in your yard, it's out in your garden, and it's in the form of wiregrass. So that's the grass that you start to pull it out because you're weeding your garden, and it's connected in a million spots on the soil. And so you keep popping up sections of roots, and then it just keeps going and going and going. And it's on a runner, essentially. And so what's happening there is there are nodes along the rhizome that will send roots down and shoots up. So we've got a vegetative structure coming up from the node for photosynthesis, and then the root stock that's going down into the soil. If we sever the rhizome between nodes, then each one of those nodes has the potential to become its own plant, and then it could start sending out rhizomes beyond that. Now, a quick caveat. If you have taken botany with me, you've heard this before. If you're going to take botany with me, you'll hear it again. But different names for different structures, for different techniques, vary regionally. So a technique that we use here in Texas is going to likely be called something different than in New England or in Old England or in South America. So it's important to know what some different names are and be ready to have a conversation with somebody at a garden center or in a forum online where they use different terminology than you do. But if you can describe the technique or if you can describe the plant part, you're gonna get yourself somewhere, and then you're gonna learn some new vocabulary. So that being said, let's look at this image. Here we see a very clear illustration of a rhizome. And maybe now that you're seeing it, it's something that you can kind of recognize or visualize. And what I want to point out with the terminology is often when it is growing underground, that's when we call it a rhizome. Colloquially, if it's growing above ground, that's when we call it a stolon. A great example of a stolon is on a strawberry plant. Now, oftentimes we will just go ahead and refer to these as runners in a colloquial setting, but on a slightly more scientific level, we would definitely call that a stolon. So what we're seeing is this above ground stem that starts to grow new nodes with rootstock and vegetative stock off of each node. And then when we, like I said before, when we sever the stolons between those nodes, then we have complete independent plants. So in addition to strawberries, some common examples of plants that propagate from stolons are mint, spider plant, and Bermuda grass, also known affectionately as wiregrass. A little tidbit of advice that I like to include in all of my classes is using technology to our advantage to educate ourselves. You're already doing it. Congratulations. But educate ourselves on using our own research as well as learning how to use Google and Google search and Google images to identify plants and help to familiarize ourselves with plant structures, plant ID, different plant parts. So I want to take a moment really quickly to give you an example of how I like to do that. For someone who loves being outside and loves plants, I also have found myself living on Google Images, whether it's pest identification, plant identification, whatever the case may be, this becomes an amazing rabbit hole to fall down. So here I did an image search for stolons, and I've got a lot of great examples of them. So here is that famous strawberry stolon from biologywise.com. This is another really great image of stolons growing on grass. Let's see what else we've got here. So I think, yeah, this is an example of a spider plant. A lot of us have this hanging out at the house, in the office. This little guy is the plantlet that's growing on the end of a stolon. And so all you have to do is give it a clean cut right there, and then put this little guy down onto some soil, give it plenty of water, and there you go. You got a new plant. This here is an example of the ryegrass, some sort of ryegrass. And what we can see here is at this node, it's starting to send a little tiny root down. We can all see that there together. Moving on to tubers. Tubers are thickened rhizomes or stolons. So this is when the stolon or rhizome no longer is exclusively for moving outward to propagate on a larger area, but it becomes a function of starch storage for the plant. So our examples of that are carrots, shallots, taro, ginger, yams, turnips, cassava, also the potato that I rescued off of the compost pile. I have some examples of tubers right here that I was at my friend's house and she had some amazing plants growing, and I decided that I needed to take some home. So this one right here, I believe is turmeric. And when I first grabbed it, I just stuck my fingers into the soil. I grabbed a chunk out of it, and then I think that was about 2 months ago, and it has already started growing all of these roots. So eventually, when I put it back, oh no, I gotta get it back in the jar now. When I get it into some soil, it's going to start to expand and propagate itself from there. And so as that tuber gets bigger and develops and stores more starch for the plant, which will help with the process of photosynthesis and nutrient movement and exchange, then we would be able to break that off into more pieces and then propagate each one of those beyond from there. Now we talk about bulbs. Botanically speaking, a bulb is a short stem that has been structurally modified through evolution by the plant to serve as starch storage, and that helps the plant during dormant periods. So when we look at it in gardening and in landscaping, we might also call these ornamental bulbous plants. Sometimes we just call them bulbs, but these are going to be our lilies, our onions, hyacinths, tulips, even garlic. My friends, did you know that onion and garlic are not roots? The part that we eat is not a root. That's a modified stem structure. So often when we buy our onions and garlic, our leeks, shallots, things like that, the roots have been cut off of it. Now let me tell you, as an aside, as a piece of personal advice, when you go to the farmer's market, because I know that's where you get most of your vegetables. So you either grow them yourself or you have a CSA subscription or you go to the farmer's market because we always want to buy locally. If you can, get these tubers, these bulbs with the roots still attached to them, because especially if they're grown organically, that's one of the best parts of the plant. So you want to rinse them really well, you want to get them clean, and then you want to chop them really fine, and then it's the most wonderful flavor and a little bit of crunch to use as a finishing touch on your salads or on your entree dishes. So we have here a very silly little illustration. So this is the onion that we know and love and are familiar with. It actually has these curly little roots. This is the entire root system right here that is taking up water and nutrients. And then this is the modified storage structure. This is the modified stem. And then here we have the leaves, the shoots that will actually function for photosynthesizing. So with the combination of the roots providing water and nutrients, and then we've got the shoots and the leaves that are bringing in sunlight and carbon dioxide, that all converts into starch right here in the middle. So here's another foray into our Google image search. So here I did plant bulbs, and I want to show you how you can fall into these super fun little rabbit holes. So this is one of my favorite images, which shows us, this is from a yard and a half landscaping co-op. So it shows us some different types of spring bulbs and the depth to plant them at. But then if we scroll down here, then we start to find all kinds of different, and I think this one's from Pinterest. Let's see what we've got here. So here we actually have a diagram of different types of bulbs and different depths to plant them at. So while we would all love it if I could, in this class, provide you with all of the knowledge you need for all of your gardening and landscaping endeavors, the truth is there's going to be a lot of information out there for you to discover yourself. And I really encourage you to start using the internet as one of your best resources. Now, caveat here, please make sure that whatever resources you're using are academically or scientifically vetted. So things that end with edu is going to be helpful. Things from scientific journals are going to be helpful. I very much caution you against taking advice from gardenloverdude.net, right? So, but it's also really helpful on the Google images to kind of, especially if you're a visual learner, to kind of get an idea of what things look like when they're being placed. Speaking of storage structures, up next are corms. So we can see in this image that they're the little growths on top of the existing storage structure that is the modified stem. So this is where we see on the top left corner and the bottom right corner, it is basically an equal amount of modified stem. But in the opposite corners, we see these little buds, they almost look like. Sometimes, in some cases, these structures can be used as propagative material, but most often they just do storage of either starch or water. We've talked a little bit about plantlets and pups already. So the top image is of the spider plant, which we already looked at on our Google image search. So these are the small plants that will develop either on the end of a runner or at the margin of a leaf. So when we look at the bottom image, maybe you recognize this one. It's pretty common in this area. This is going to be the mother of thousands or mother of millions. And millions is actually, I feel like, more accurate. So each one of the plant's leaves or lobes is lined with all of these tiny little adventitious plantlets. So we can see that the white strings coming off of the bottom of the individual plantlets, those are roots. So as soon as it hits a solid surface that is not its parent plant, it is going to take root, and they go crazy. This plant is almost a weed. Now, as I mentioned before, asexual propagation is also cloning. And the reason being is that it has the exact same DNA in the parent plant as the propagated plant. Let's go ahead and talk a little bit more about that. Just as with animal cells, every cell in a plant contains all of the genetic characteristics of the entire plant. And so that's what helps us with asexual propagation, is because each plant part has the ability to potentially grow all of the other missing parts of the rest of the plant. So for example, taking a leaf off of a snake plant can result in growth of roots, because with the snake plant, all we have are leaves and roots, right? Let's look at a snake plant real quick. As usual, we look it up together. So snake plant images, we know this one. This is our snake plant, also known as stepmother's tongue, also known as sansevieria. Like I said, when we take the leaf of a snake plant and put it into soil or another propagative medium, then that leaf will start to grow roots. The same thing goes with growing a plant from rootstock. We take a chunk of the root and then it grows the rest of the corresponding plant parts. So the plant parts can either grow a new root system, a new shoot system, or both. So let's go ahead and look at the advantages and methods of asexual propagation. First and foremost, as we mentioned, plants that are propagated asexually are either genetically similar or, in more instances, genetically identical to the parent plant. Our seed propagation, sexual propagation, comes with genetic variations. Now, this can absolutely be used to our advantage when we are trying to create new hybrids of plants. So what we get is a combination of genetic material from the two parent plants. So if we look back to our high school, hopefully, genetic lessons, this may be something that we remember. So a quick review. We remember this? So we have parents A and B, and then we have our offspring here, and we put that as lowercase a and b, or AB. So when we take two sets of genetics in the parent material and we cross them through sexual propagation, then we end up with a new blend that's a little bit different. So we also remember that in genetics there are dominant and recessive traits. Typically, dominant traits will show up more often in the following generation, but it doesn't mean that the recessive traits disappear entirely. And we see this in life, right? Any of you who have siblings. So for example, my sisters and I all look very much alike, and I am the only one with curly hair. So the same thing will go for the plants that we start to hybridize together. A lot of times Home gardeners will try this with flowers and with tomatoes. So let's talk about tomatoes for a moment. Let's say that you have two different types of tomatoes that you've been growing and you've been saving your seeds and you really like that process. By the way, we have a whole lesson on how to do seed saving and create hybrids. So let's say that one of those tomato plants has the most perfect flavor and you want to find another tomato plant that has that same flavor, but it also, as soon as it gets a little bit cold, it stops flowering and the vines die. And then you have another tomato plant that has a decent tasting fruit to it, but what you love about it is it can get down to 40 degrees and then it'll still kick and it produces tomatoes later in the season than your other plants do. So why not try to combine those genetics to get the best of both worlds? So that is one of the advantages of doing asexual propagation and hybridization, but it's a gamble, especially if you're starting from scratch. You don't know if you're going to end up with a tomato that has mediocre flavor and dies as soon as it gets below 60 degrees, or are you going to end up with the perfect hybrid? It takes a lot of experimentation and trial and error. And with plants, experimentation and trial and error can take a long time because if you're growing from seed, you have to start from seed. On that note, that's one of the reasons why asexual propagation is preferred, because not only do we know exactly what genetics we're going to get out of the new plant, but we also don't take as much time to figure out what the genetics are going to be. And if we are trying to maintain integrity in the genetics of a strain of plant, this would allow us to maintain that cultivar for infinite generations. Another advantage to consider is not all plants produce seeds, or even if they do produce seeds, sometimes they're not viable for germination. So plants that don't produce seeds are going to be our ferns, mosses, liverworts, and anything in the gymnosperm family. And then there are the economically significant fruits that we don't grow from seed. For example, seedless grapes. Obviously, in that case, the only option is asexual propagation. Same for seedless oranges. And then the bananas and strawberries, we see the seeds on the fruit, but those are not mature enough to be viable for propagation. So those plants are asexually propagated. Now, side note, with strawberries, that's pretty much totally okay because as we saw earlier, they create runners and they propagate themselves on stolons. Then there's also our indoor plants that typically just are used for their foliage. They don't reach maturity to flower. And so those examples are snake plant, like we looked at a moment ago, and ivy. As I mentioned a moment ago, growing plants from seeds is starting from scratch. So when we use asexual propagation, we have the advantage of a faster method of growing plants. It takes less time than growing plants from seeds. A lot of seeds require a good amount of time to germinate, and then it takes an additional amount of time for them to establish themselves and then eventually reach maturity. And then there are even some seeds that require periods of rest and dormancy before they germinate. An easy example for Central Texas is our wildflowers. The wildflowers drop their seeds in late summer when the flowers die back, but then they don't germinate again until the following spring. It's also important to note that some seeds have hard seed coats and they will only germinate when the conditions are favorable in their environment. And then there's the teeny tiny seeds that are difficult to handle when they're being planted. So some examples of seeds that don't germinate without those environmental stimuli. One is good for this area, the chili pekin plant. So that's the pepper plant that has the teeny tiny super hot chilies on them, and they typically perennialize. So they will go dormant but survive through the winter and then fruit and flower the next spring. Those plants are very, very difficult to grow from seed because their seeds have a hard coat around them that needs to be broken down with some form of acid. And the perfect form of acid exists inside the stomachs of birds. So the bird eats the pepper and then passes it, and after the seed has been broken down and then deposited in perfect growing medium, we'll talk about more of that later, then it will be ready to germinate. Another example of that is going to be a lot of coniferous trees. So those are going to be our pines and firs and things like that that have cones. A lot of times the cones will only open and release seed if there is a fire. So either from the heat of the fire or the chemicals in the smoke of the fire will cause the cone to open up and then release those seeds. Unfortunately, Smokey Bear has made all wildfire bad for all of us, whereas it was actually it was and is a very important part of the ecological process and the life cycle of a forest. But that's another soapbox for another class. Our conversations around advantages and disadvantages will often come to the amount of time that it takes to grow a plant. This is significant because if you are trying to grow plants for a profit, time is expensive. Time is your most valuable and least renewable resource. So if you have plants that need space in your greenhouse in order to germinate and then grow to maturity, they are taking up space that you can't put other plants in. So the longer those plants sit there, the more expensive they become. Some plants take a really long time to germinate. Best example is growing parsley from seed. It can take parsley up to 21 days for germination, and then they remain really small. And it takes a long time for their rootstock to develop to maturity to be able to take up enough nutrients and water to start to grow a larger amount of above ground vegetative mass. So as we think about not only the growth and the development of the seedlings is fairly slow, once we do have those seedlings, they are tender, delicate little babies. And they take lots of additional care. They take lots of nurturing. It takes a while for them to establish themselves to maturity to be able to grow heartily without needing that extra babying. So when we use asexual propagation, we take a part of a plant that has already reached maturity and we make a whole new plant from it. Asexual propagation allows growers to bypass the juvenile characteristics of certain plants. There are a lot of flowering and fruit trees that, when grown from seed, they go through a juvenile period before they produce flowers and fruit. There are some trees that take 5 to 10 years before they start flowering the first time. So not only are we losing control over what those fruits and flowers come out looking and tasting and feeling like, but we would also have to go through the entire process of many years to find out. So, as an example, trying to grow your own fruit tree from, we'll say, a peach pit or orange seeds. It's going to be a good number of years until you actually get an edible fruit off of that tree. And then, even so, you don't know what it's going to taste like because you don't know what the parent material was and what kind of pollen went into the flower to make that fruit. My friends, the advantages continue. Another advantage of asexual propagation is that growers are able to produce plants that are adapted to the soil and climate of a specific area. I don't know if you know this, but it is difficult to impossible to grow blueberries and avocados here in Central Texas. So that would be an instance where we might take a plant with a desirable flower or fruit that has naturally a weak root system that might be susceptible to pest. They aren't able to grow in a certain area or in a certain type of soil. And then we would use a budding or grafting technique so that we would take the part of the plant that creates a desirable flower or fruit and then graft it on to a compatible plant that has strong rootstock that is able to survive and thrive in whatever climate the fruit and flower wouldn't be able to grow in on their own. Asexual propagation, budding, and grafting will create an opportunity for growers to control the size and form of the plants that they're growing. And my favorite example for this are dwarf fruit trees. So the rootstock is going to be in control of how the above ground shoots grow. And that is going to keep it in dwarf form, which is desirable for harvesting. I don't know if you have ever tried to harvest the fruit off of a full-size tree, or if you have a friend with an orange tree or a Meyer lemon tree, maybe you have one, but often at the end of the season, there are some unreachable fruit at the top of the tree. And so if we have a dwarf variety, it makes it easier for us to clean the whole tree off. Now we get into a rundown of the basic methods for asexual propagation. We've got cuttings, we've got layering, we've got division, budding and grafting, and tissue culture. Let's look at those individually. The first is cuttings, and it's exactly what it sounds like. We cut any part of a plant from the parent plant, and then we try to grow the rest of the plant from the cutting that we took. So in order for it to be successful, the plant part that we cut off has to be able to produce all of the missing parts from the plant. So a stem cutting has to be able to develop new roots. Cuttings from leaves need to be able to develop root and shoot systems. Cuttings from roots have to be able to produce the rest of the above ground vegetation from that piece of root. This here is an example of growing from a cutting. So this here is one of these beautiful hangy, drapey, sort of viney indoor plants. I believe we call it a money plant. But again, it's all regional. So my friend Marissa from the Sustainable Agriculture Department, she has a huge one of these on her porch. And several months ago, I asked her if I could take some cuttings, just got some clean scissors, snipped it at a 45-degree angle. Don't worry, we'll go over this. We have a lab for it. And then I just stuck them in water. And now look at the fantastic root system that it has developed. So this is a perfect example of a cutting growing the rest of the missing part of the plant. Up next is layering. So just to be clear, I'm doing a real quick introduction to each of these different types of propagation techniques, and we will go into them more in depth as the course continues. So when we do layering, we root one part of an existing plant, and then once the root system has developed in that rooted layered part, then we sever the connection between them. So essentially what we're doing is we are creating or simulating a stolon and then forcing rooting to happen at a node that we create ourselves. So there are two different types. There's air layering and ground layering. In this image we see here, this is ground layering. And it works really well for plants that are close to the ground and have at least somewhat flexible shoots so that we could lean one over onto the surface of the soil without snapping it off of the main plant. For plants that we can't do that with, and that would most often be trees, we take a technique called air layering. And I want to give you a cliffhanger on that and leave you at the edge of your seat, because we will come back to air layering later in this course. Now we get into the method of division, where we literally just divide different parts of the plant away from one another and allow them to be independent plants in and of themselves. The example that we have here in this image is the snake plant. So what we can see is each separation, each division has a shoot and a root, and that's all the plant needs to be able to continue to live and then to develop additional vegetative tissue. So this is going to be really common for plants that grow from bulbs, corms, and rhizomes, but we also see this in houseplants. So this little guy is a perfect example of propagation by division. So if we look down in here, we see that we have all of these individual shoots. So what we're going to end up doing, and we'll do it together in this course, is we're going to take all of this out of the pot, and then we're going to very gently separate the rootstock for each individual shoot away from one another, stick them into new pots of their own, and then over the next few months, it'll fill back out just like this. And so you just have this continuous supply of new plants to give to your family and friends, and you can fill everyone's life with the joy of plants. Now, budding and grafting is a very exciting procedure that involves joining separate plant parts together. So it's successful when those plant parts become a union and then they start to grow together. So what this comes down to is a couple of things. One is the plants have to be botanically similar enough to join their tissues together. And the second is there has to be a compatibility in the vascular system. So the xylem and phloem need to be able to line up in such a way that there is an exchange of photosynthetic materials and water and nutrients. It's the same idea as, well, I don't know, I'm not a doctor. I play one on TV. But if we were to sever a finger and then have to sew it back on, the surgeons would have to connect each individual artery and vein for that finger to be able to survive and succeed on the hand. A very cool and intimidating to me type of propagation is tissue culture. So we take a small piece of plant tissue and then put it into a specialized medium in a totally sterile environment. So in a test tube or in a petri dish, and then the tiny bit of tissue will actually develop all of the other missing plant parts. So we would be able to clone an entire plant out of a small bit of tissue. Now, one of the most important parts is that the culture and the medium surrounding the plant tissue has to remain absolutely sterile and avoid contamination. Now, I've never pulled off tissue culture for a number of reasons. One being, it is really hard in my life to create and sustain a sterile environment. We know what it's like having pets. But the other reason is that the tissue medium, the agar that we would need to put it in, is much more specific than just the average potato dextrose agar that would be used in a high school or college biology lab, right? So the coolest thing about this type of propagation is it can literally save the genetics of a plant if it's contracted a disease. In the last year or so, we've all learned so much about viruses and the nature of viruses and how they work. And one of the things that we have learned, if we didn't already know it, is that viruses attack individual cells and they replicate their own DNA inside the cells of an organism, and then they spread from cell to cell. So, unlike a fungus, for example, powdery mildew. Powdery mildew is going to largely grow on the surface of a plant, and we can either, if we catch it early, we can clean it off and stop the infection from there, or we can clip that leaf or that shoot and sever the infection from the rest of the plant. But with a virus, once it gets in there, if it has the opportunity to spread all the way through the plant, it's over. The plant's DNA has been irrevocably altered. So, this tissue culture method is an opportunity, if we can catch it, to sever the uninfected part of the plant and then clone it from there. So, especially in the case of like an heirloom variety of plant or some sort of, I mean, there's a lot of high dollar house plants out there. And if they were to get infected with something, this would be the only way to save that genetic integrity. So let's look at what it looks like in a lab. Here we go. All of these plants that are sitting on that sort of clear gelish substance, that's not water, that's a special kind of agar medium. All of these little plants and the healthy root systems we see below them were grown from just a piece of tissue. Now we're going to very briefly go over sexual propagation because we have an entire lecture devoted to propagation by seed. So very simply, we may remember this from grade school, but there's the male parts of the flower and the female parts of the flower. The male part is the stamen, which is made up of the anther and filament. It has the pollen, that the equivalent in an animal body, the pollen acts as the sperm, essentially. Then we have the female parts of the flower. So that is the pistil, which is composed of the ovary, the stigma, and the style. So when the pollen comes in contact with the female part of the flower, the pistil, the pollen fertilizes the ovary in the plant, and that will eventually develop into a mature seed. Now here we see a very simple illustration of the anatomy of a flower. So we all know that the petals are those pretty pink things. And then when we look at the very bottom, that's the receptacle. The little wings or arms, the green ones coming off of the sides of the receptacles, those are the sepals. The sepals are the protective layer of tissue that will surround the bud before it opens up and blooms into a full flower with its petals exposed. So those sepals are there to be protective against the environment and from any potential pests. Collectively, the sepals are called the calyx. Then above and between those sepals, we see the ovary. So this is the base of the organ that collectively is called the pistil. When we move up that pistil into the thin neck of the structure, that's called the style. And then at the very top, we have the stigma. Now the stigma is sort of a sticky landing pad for grains of pollen. And I say sticky because it really is. And different plants, different flowers have a different stickiness to it depending on largely how large and heavy the grains of pollen they're trying to catch are. The bigger and the heavier the grain of pollen, the stickier it needs to be so that it doesn't just bounce and roll off of the stigma once it comes into contact. So it is waiting to receive pollen from the anthers, these little yellow guys. There's 5 of them on this picture. The anthers are attached to the filaments. The filaments are just a prop for the anther, which holds the actual grains of pollen. And there we go. This has been our fairly rudimentary introduction to plant propagation. So as we move through this course, we're going to talk about each of those things much more specifically and much more in depth. And I will warn you, there's a lot of repetition in this class. It's part of one of my philosophies as a teacher is that the more we hear something, especially if we hear the same thing used in different examples and through different perspectives, then we really start to be able to internalize and conceptualize and really come to grips with some of these concepts. And some of them will be easier than others for some of us. Science is in a lot of ways until you really grasp some of the main concepts, science can actually be fairly counterintuitive. So I just want to warn you that I come back and I repeat myself a lot of times because that is how the most important points are going to sink in a little bit better for you. And I've said it before, I'll say it 100 times. One of my objectives as your instructor in these courses is to teach you how to be able to think about plants in the correct way to be able to do research yourself. Like I said at the very beginning of this lecture, I would love it if in 32 hours of continuing education class, I were able to impart on you everything you would need to know. But in reality, that's not how it goes. So my objective is to give you the information you're going to need to be able to do the critical thinking that it will take to do research to get you to the answers that you need for your own personal journey with plants. Thanks for hanging out with me for this period of time, and our next lecture is waiting for you.